The organization of metabolism unfolds in two major interconnected processes, catabolism and anabolism. Catabolism is responsible for breaking down complex molecules into simpler forms, releasing energy that the cell can then use, primarily in the form of ATP and reduced coenzymes such as NADH and FADH2. These pathways are degradative and oxidative. As substrate molecules lose electrons, those electrons are transferred to coenzymes. In contrast, anabolism is the process of building complex molecules from simpler ones, which requires an input of energy, mainly ATP generated by catabolic pathways, and the reducing power of coenzymes like NADPH. Anabolic routes are biosynthetic and reductive in nature, and they are essential for constructing the cellular components needed for growth, repair, and a variety of vital functions. The energetic relationship between catabolism and anabolism is critical. Catabolic pathways act as providers of chemical energy stored in ATP, NADH, and FADH2, and this energy is then used by anabolic pathways to convert small precursor molecules into cellular macromolecules. Within the cell, the numerous chemical transformations catalyzed by enzymes can be grouped into 6 fundamental types of metabolic reactions. Oxidation, reduction, redox involves the transfer of electrons from one molecule to another. Ligation forms covalent bonds, such as carbon-carbon bonds, and requires the energy from ATP hydrolysis. Isomerization rearranges atoms within a molecule to create isomers. Group transfer moves functional groups, for example, methyl, phosphate, from one molecule to another. Hydrolysis breaks chemical bonds through the addition of water. Additional removal of functional groups modifies molecules by acting on double bonds, adding or removing functional groups. For many enzymes to function correctly, they require cofactors, either metal ions or coenzymes, organic molecules derived from vitamins. Cofactors act as carriers of atoms, electrons, or functional groups. Coenzymes in particular can be classified according to their roles. Electron transfer coenzymes, NAD+, oxidized form, accepts electrons during the degradation of fuels. Its active site is in the nicotinamide ring. When reduced, it becomes NADH. FAD, oxidized form, accepts electrons via its isoalloxazine ring, and its reduced form is FADH2. NADPH is mainly used in reductive biosynthesis, donating electrons. Unlike NADH, which focuses on generating ATP, NADPH provides reducing power for anabolic reactions. Acyl group carrier, coenzyme A, CoA, transfers fragments of 2 or more carbons. Its reactive thiol, SH group, forms thioester bonds, for example, as acyl-CoA. Phosphoryl group carrier. ATP is not only the primary currency of free energy in the cell, but also serves as a coenzyme that transfers phosphoryl groups to drive various cellular reactions.